Well, so greetings everyone. It's my great pleasure to introduce tonight's speaker in the presidential lecture series in the area of emerging diseases and infectious diseases, Dr. Peter Daszak. Before I do that, I'd like to take a moment and note that Peter's presence here is actually part of the university and the president's support of a PhD training program that we have here at the university on the ecology of infectious diseases. And part of that support is allowing the PhD students to identify and nominate, invite, and help coordinate the visit of speakers like Dr. Daszak here, who has great recognition in this area of emerging diseases. So I think it's actually a great opportunity for us to have him here. Peter got his bachelor's and PhD degrees in the UK, actually, as a bachelor's of science in zoology, and followed by a PhD in parasitology at East London University studying coccidia. He then came to the US to work with the CDC on the Nipah virus outbreak, and then on to the University of Georgia working on the amphibian decline problem, and was one of the people, the collaborators that identified chytridiomycosis as the culprit behind amphibian decline in the US. He now serves as executive director of the Consortium for Conservation Medicine in New York City, and also has adjunct appointments at Columbia University and at Tufts. So with that as a short introduction, I would like to let Peter take over.
Well, thank you very much. It's a great honor to be here. Thanks. And once again, thank you to the President and the staff and the students especially for the invitation. What I want to talk about today is what I consider one of the biggest problems in global health right now. And by global, I mean our health and the health of people all across the world. Emerging diseases are diseases that are moving into our population for the first time from other animals, or diseases that are increasing the scope. So they're a big threat. And we think of diseases like SARS, Ebola. They're diseases that cause very high mortality, but don't really have very high mortality rates, but don't really cause that many cases every year. But if we think about HIV or influenza, we see diseases that move rapidly into our populations and persist and cause very extensive problems, both in terms of death, morbidity, and economic loss. So one of the sort of holy grails of this field is to say, can we get to a stage where we understand this process of disease emergence to the point where we can predict it? And if we can predict what's going to happen, we can then get into a situation where we can control what's going to happen and hopefully avoid some of the devastating outbreaks. And that's really what I want to talk about and go through some of the examples that our group's been working on for the last few years. If I can work the slides. It's important to realize, of course, that emerging diseases, this new group of threats, is nothing really new. And when we think back through human history, we've been seeing this sort of event repeatedly throughout our history as a civilized community of animals. If we go back to biblical times, the Old Testament biblical plagues, there is really good evidence that those things actually happened. It's just at the time when we were domesticating animals from the wild, sheep, cattle, dogs, we're bringing them into very close contact with our populations. And we were also building large civilized communities where we had a density that was able to keep this level of disease transmission high enough to cause problems. And if you look at the genetics of, say, measles virus or smallpox, it turns out that the closest ancestors of those viruses historically were diseases of cattle. So domesticating them led to their spillover into our populations and the evolution of a new pathogen, which became measles or smallpox. And as we go through human history, we then move into a phase where we globalized for the first time, when Europe reached across into Asia and set up the Silk Road and brought with it bubonic plague, the Black Death, and which eventually killed probably a quarter of the European population. And then later, when we globalized even more, when the Western cultures who'd now developed an endemic relationship with diseases like measles and smallpox, then colonized the New World and took those diseases with us and introduced them. People like Hernan Cortez here in the picture introduced those diseases into what are essentially naive populations of people that had never experienced those diseases before. The result were devastating outbreaks and almost extinctions of local native Americans. But of course, it doesn't end there. We then move into the industrialized era and once more globalization in terms of the First World War, and probably one of the most famous outbreaks of all time, the outbreak of so-called 1918 flu, the Spanish flu. Didn't really come from Spain, but in fact was transported across with our troops from the US into Europe and back. And if you look at the mortality rates, probably killed more people during the First World War than actually died from bullets and bombs. So diseases can be truly devastating, but they are a part of everything we do. And what I'm going to try and show today is that this new phase of disease emergence is also a part of what we do as people on the planet. Not only is it a part of our sort of life history, it's also a part of our culture. I just want to take a little moment to show how we almost celebrate the impact of diseases. This is the famous book. I mean, one could almost take a disease tour of the world, and I recommend a couple of places in England where I'm from. We had some great diseases in the old time, and this is bubonic plague. The village of Eyam, the so-called plague village in Derbyshire, which was one of the last towns to ever be hit by the bubonic plague in England. What happened is the inhabitants of this town sealed off the town. They knew what was going on. They'd imported fleas in some wet cloth from London, started dying, so they sealed off the town voluntarily, to the point where food had to be brought in and left for them. They'd pick it up and they'd put the money in a stream, and people would pull it out of the stream. So the first realization that these things were communicable, and so that you wouldn't get infected as you pulled the coins out. And this is the Book of the Dead for Eyam, a beautifully illustrated manuscript that lists the people that died in the years 1665 to 1666. And this, you might think, is also an odd medieval curiosity. Well, actually, it's modern. These are the plans for the Whitechapel Hospital stained glass windows in London, England, the hospital where the Elephant Man came from. And there are some researchers there who want to celebrate their efforts to control infectious disease. Part of our culture again. And this stained glass window here represents the mortality curve of the 1918 influenza pandemic. And this is one of the drugs used to treat HIV. So emerging diseases are part of our history, they're part of our current life, and they're part of our future. And what I'm trying to say is, let's try and minimize their impact in the future. So, what is an emerging disease? Well, HIV/AIDS is a great example of emerging disease. It spread rapidly around the world into the human population due to bushmeat hunting in Africa, and then through global air travel and demographic patterns and human behavior in the West, causing a devastating outbreak. And even with the HIV drugs, we're still looking at a mortality of 40 million people, a very large outbreak. SARS, the disease that hit the headlines as the first pandemic of the new millennium. West Nile virus, a disease I know very well. I live in New York now, and the mosquitoes in my yard carry West Nile virus. The birds are infected. We find dead birds that died of West Nile virus. And some diseases that some of you here I know very well. Hantavirus, mad cow disease, a case in Montana that just one single infected cow cost many millions of dollars to the cattle industry in just overnight impact. And chronic wasting disease that's causing problems across the West. These diseases are emerging in that they're caused by pathogens that are on the move. They infect new hosts, so they're jumping from animals into people. They're expanding geographically, like West Nile. They suddenly appear in a new place. Or they're changing the pathology, like hantavirus pulmonary syndrome. It's been around for a long time in various guises, but here's this new type of pathological impact. Or they're evolving. And when we think about drug resistance, what better example of evolution in action? We use a drug, we select the pathogens that resist the drug, and we have drug-resistant microbes. And now we're getting to the level where we have pathogens that are almost resistant to every known antibiotic class. So obviously, diseases like HIV cost a lot of lives, but also they have a huge economic impact. If we look at this graph, it's a plot of the economic impact in billions of dollars versus time. For instance, Lyme disease here, $2.5 billion, but actually spread out over many years. Foot and mouth disease in the UK, one single outbreak costing $20 to $30 billion. Or SARS, one single outbreak globally costing between $50 and $100 billion. And some of the estimates for H5N1, avian influenza, if it goes pandemic, if it successfully moves people to people around the world, maybe as much as $350 to $800 billion in 1 year in a single outbreak. So this is a very significant impact, almost as much as the current economic crisis. It's the same order of magnitude. But I want to just take a moment to talk about how I got involved in this field. I've always had a passion for wildlife, and I got involved in, as an undergraduate, I wanted to do a research project on reptiles, but unfortunately the only one left was to work on reptile diseases, so I sort of got thrown into this disease side. But when I started working with a colleague of mine, Andrew Cunningham at London Zoo, we found a whole new series of diseases in the zoo collections and in wild populations, including in amphibians. And amphibians have been declining globally for the last few decades. And this gives you an example of what happens in these population declines. This is year by year in Panama, and these are population captures, the number of animals captured per person per hour in these transects that people run year by year. And you can see it looks like these animals are just dropping off the cliff. These populations are crashing suddenly across a whole series of sites to almost zero. Now, what causes that? At the time, people really didn't know, and there were all sorts of theories put out there like ultraviolet light due to the ozone hole, chemical pollution, climate change. Well, actually, it's an infectious disease. We know that because a couple of ecologists had the common sense to talk with veterinarians and cross the disciplines, just like we're trying to do now in the consortium that I run. This is one of the Australian frogs that first allowed us to identify this pathogen. And actually, this is a dead frog. And this reminds me of the Monty Python sketch where John Cleese goes into the shop and says, "Excuse me, sir, my parrot's dead." "Well, no, it's not, it's resting. He's having a little sleep, sir." Well, this is a sleeping frog. And we know that because when they're alive, they normally look like this. They're very, very ubiquitous, if you get my drift. If you look carefully, you'll see the only real sign that this frog is in any trouble, other than it being dead, is that it has a thickening of the skin. And it took many months for the pathologists that we work with to work out what was going on. When you cut a cross-section through the skin, now amphibians breathe to some extent and take in water through the skin. So that skin layer of keratin cells needs to be just one cell thick. Here it's many cells thick, and it's got some curious bodies inside. They're fungi. And you can see on this scanning micrograph, each cell has a little bulge with like a bowl-shaped fungal spore. It's a raging fungal infection of the skin. Now, for a frog that breathes through its skin, that's a problem. And in fact, it's a new genus, a new species of pathogen, given the awful name Batrachochytrium dendrobatidis. It's so awful that everybody calls it BD right now, so it's a lot easier. It's the first time this group has ever been shown to infect vertebrates, and it was causing frog declines globally, and simultaneous emergence in Australia, Europe, Central America. So how can this be? How can a disease of wildlife suddenly emerge globally, just like SARS did? Seems very odd. Well, it turns out that we have a global trade in frogs. We like to eat frogs' legs in our Chinese or Thai restaurants. And if you ever do that, it's usually bullfrogs in America. It's not even bullfrogs caught locally, it's bullfrogs bred in Asia or Latin America, shipped back live into the US and butchered locally. So what a great way for a disease to get spread around. You take a frog from North America with a disease, take it down to South America, it infects everyone there, and then you start shipping them back to North America and spread the disease around the world. And we've now found this pathogen in the food trade for frogs' legs, in the lab animal trade, in the zoo trade, in the pet trade, and in species that have been introduced. And this gives you an example. These little grey things are actually frogs. This is a frog farm in Taiwan with something like 1 million frogs at any one time. And these are bullfrogs, North American bullfrogs, shipped back over to the US. Now look at this water system. The water gets sluiced in right out into the local river system. What a perfect way to spread a disease. I've just got to add that, everybody who knows the history of England knows that there's a certain animosity between the French and the British. And in England, we refer to French people as frogs because they eat frogs' legs. Well, embarrassingly, so do the Brits. And we got this data from the UK government records. Frogs' legs, fresh, chilled or frozen, come and get them. £1 a dozen. Well, embarrassingly, if you look at the quality of the British Customs and Excise Office, every time we count the annual number of frogs' legs coming into the country, we manage to get an odd number. So I think something is wrong there. Anyway. So I think that the wildlife are telling us something. As we trade animals around the world, we spread diseases, and just like we did with SARS. What's the impact in wildlife? Well, one of the impacts is extinction. If you go to Hawaii, if you could have gone to Hawaii perhaps 500 years ago, you would have seen this amazing array of beautiful honey creepers. You can still see the EV here, but sadly, the green ones are extinct and the blurry blue ones are endangered. Over 75% of this group of endemic birds has died out due to introduced pathogens that we brought over. We brought the mosquitoes with us on ships, and we brought the pathogens over with us and introduced them into Hawaii, and they've caused the extinction of birds out there. So, seeing large populations of wildlife die off due to disease is not unusual, just like it's not unusual to see diseases spread around the world during the World War I. So it's also not unusual to see SARS or HIV or pandemic influenza spread around the world. I've personally had an experience of this extinction event, and I think two very sad stories. One is these beautiful gastric brooding frogs. This is a species of frog that was only discovered back in the 60s. The male frog eats the eggs, secretes an anti-acid product that stops acid secretion in the gut, and then the little baby frogs develop in the stomach, and it spews out little froglets a few weeks later. And there's one in the act of spewing out a froglet. Well, that's very nice. And in fact, the drug companies were hoping to get some sort of ulcer drug out of this, because a drug that blocks acid secretion would be very useful. Unfortunately, it's extinct. It's extinct probably due to this infectious disease that we discovered in other frogs. And here's my finger here with the last of a species. This is Partula turgida that died out on January the 1st, 1996 in London Zoo. It's a boring snail, but it's also an extinct snail, and it died out due to a disease that we introduced into the zoos that spread through the collections and wiped them out. So diseases can have a big impact. That's the message here. And if we have these global travel networks that we spread diseases through, what about the global travel of people? This is a map of just 1 year's aviation. This is numbers of people traveling. The brighter the shade, the more travel we do. And of course, the US, Europe, and some of the richer countries of the world, that's where we do the most traveling. So if you think about it, whenever a disease emerges, no matter how remote, if it has the capacity to get into this human travel network, where's it going to go? It's going to gravitate to where we are. So I think the message here is this really is something to worry about, not just globally, but also especially for us, because these diseases will gravitate to our populations. And that level of travel is increasing. This is great news if you've got shares in Boeing. I got these data from Boeing website. They do an annual share report. They analyse the global travel of people around the world. It's their business. This is the number of people travelling, the pink bar increasing every 5-year period up to the year 2000. The blue is the number of thousands of kilometres of new travel routes, aeroplane routes, globally. So we're still globalising the planet. And they predicted over many years that this travel share of GDP will continue to increase for the next 20 years. And I've gone back and checked their predictions, and they're accurate. We value travel extremely highly. It's essential to our global economy. And one of the costs of this travel is the spread of diseases. Okay, so what can we do about this? It's okay to whine on about it, but what can we actually do to stop it? Well, I think we can predict what's going to happen and then go out and try and put measures in place to stop these things. And what we need to do is to look at this system as a sort of three-tier system. There are some diseases like HIV, SARS, influenza, which make the jump into this global network and travel around the world. They're the pandemic diseases. They're the ones we've really got to worry about. They're the ones that have human-to-human transmission and move around the globe. But if we can go back a step before that, there are lots more diseases that never really quite reach that. They emerge in small groups of people, occasionally get big outbreaks like Ebola or Nipah virus, which I'll talk about in a minute. Maybe if we can predict those and start to act on those before they go pandemic, we'll save ourselves a lot of mortality and economic cost. And even better, why don't we go to wildlife? Since so many of these diseases come from wildlife, HIV is really a chimpanzee virus that by a curious quirk of fate got into the human population and happened to be very lethal. If we can go back to wildlife, look for those pathogens and stop people butchering wildlife or getting infected some way from that initial species, maybe we can prevent them even before they start. So that's the aim. How do we do it? Well, here's how you can predict something. If we look at West Nile virus is a virus that should be in Africa and Europe. For the past few million years, it's circulated between Europe and Africa. And then for some curious reason, in 1999, it jumped over to New York. Now, why is that? Why pick New York? Well, of course, New York is one of the biggest ports in the Americas. We have more aeroplanes landing than just about anywhere else, and we have lots and lots of airplanes coming over from Europe. So the likely explanation is that it came over in a mosquito, an infected mosquito, or perhaps some birds brought over for the pet trade from Europe. So what's happened is we've increased our air travel and reached a threshold where the virus can find a way over. So where's it going to go next? I mean, let's try and use this to our advantage and try and stop it moving to its next destination. Well, here's what we came up with in our lab. And I had a postdoc who's a real mathematical genius, but he also likes tropical islands. And he said, well, it could get to Hawaii. There's some very endangered birds there. Let's have a look at how it might get there. So we did that. We got a nice trip to Hawaii. We did the analyses. Then he said, well, what about Galapagos? I thought, yeah, that's a very good point. There's the Darwin's finches. So then we got this fantastic trip to Galapagos. We did a risk assessment with the government there. And then he wanted to work on Barbados, and I said, okay, that's enough. He did the work, but we didn't get the trip. But there's a serious reason for this. Of course, if West Nile gets to Hawaii, less people will want to go there for tourism. You don't want to catch West Nile when you go and lie on a beach. And if people are going for ecotourism to the Galapagos, which is the third biggest earner of foreign GDP in Ecuador, you go there to see Darwin's finches. It'd be really unfortunate if we introduced a disease that wipes them out. So that's a serious issue. How do you predict it? Well, there are only really 5 possible ways the virus can get there. Either through infected people, through mosquitoes that are infected and get blown over, through mosquitoes that hitch a ride on boats or planes, or through people taking birds, poultry or pet birds, or through birds that migrate. Now, these all seem a bit odd, but when I flew over to Hawaii for our meeting with the government there, the state government and the Navy and the USDA, on my plane there were 3 or 4 mosquitoes and a parrot. So this thing really does happen. So what we did is we got data, we got the numbers of all of these things, we worked out the math. Very simple. And I looked at 2 subspecies of tourist, are they the risk? So we had 2 subspecies, the American tourist, Homo migrans variation americanus, and the British tourist, Homo migrans vulgaris, there with his Union Jack shorts, a classic. The good news is we're at very low risk. By the way, that isn't me when I had hair. When we get infected with West Nile, we don't produce enough virus to infect a mosquito that bites us. So we're a dead-end host for West Nile virus. This is the amount needed to infect a mosquito, and that's the amount we get. So we're of negligible risk. And in fact, the biggest risk, and this is a log scale, so each step is an order of magnitude 10 times higher. The biggest risk is mosquitoes hitching a ride on an airplane. There are just so many airplanes going to Hawaii and Galapagos now, and the average number of mosquitoes on a plane is 1.55. People have worked this out. You just simply do the math, and it's an incredibly high risk. Now, you can even work out the number of infected mosquitoes that will land on Hawaii each year for the foreseeable future, and that's between 6 and 60. Doesn't sound like much, but that's every year forever. So it will get there. The likely scenario is it will get there, unless we do something about it. So we spoke to the government of Hawaii, and they have a potential way to deal with it, which is to use residual insecticide in the cargo hold of planes. You don't need to spray the cabins because that's pretty ineffective. 80-odd% of the mosquitoes are in the cargo hold, so you put residual insecticide down and reduce the risk. So here's an example of something we can predict and then deal with the problem, control it. So what about another big issue, avian influenza? We've heard about all the paranoia. How is it really going to get to the US, and can we do anything about that? So we did the same analysis, and this time we looked at every possible way avian influenza can travel, either through poultry trade, through migratory birds, or through the pet trade, people taking birds from one country to another. We got data from the Food and Agriculture Organization on every country that had avian influenza, then the next country that got it next, and we looked at how many poultry were transported from one country to another. We also looked at how many birds migrated from areas where there was an outbreak to areas where the outbreak then happened, and just did some simple math and worked it out. And this is what we found. If you look at this global map, each circle represents a separate spreading event for avian influenza. We all know this virus first emerged in China, in Asia, in the late 90s. And basically, if the circle's orange, it means poultry trade, chickens is how it got around. And then if it's purple, that means it's migratory birds. So what we've got is a virus that lives in poultry, that spread when people took poultry from one country to another, but then once it reached countries where the birds migrated across, it became available for this global transport and started then to spread through Europe. Which is all very interesting, but what about us over in the States? We're next, it's not here yet, how's it going to get here? And if we can work that out, can we stop it? Now the government were already on the case, and they held a meeting and said to all their experts, how's this virus going to get across? And the general thought was, the virus comes from Asia, birds migrate from Asia across through Siberia and Alaska, so let's go to Alaska and start testing birds, that's how it's going to get in. Well, we did the analysis, we spent a few weeks crunching the numbers, and we found that actually, although we are protected because we stopped trading poultry with countries that are infected, and there's a nice white zero risk. But look at Canada. Canada has 200 to 2,000 infectious bird days of risk. That means 200 chickens times 10 bird days each, 10 days each of risk. Mexico, the same. Brazil, the same. Now, if the virus gets into those countries, they still trade with infected countries. It's just going to jump across the border. In fact, we have a quarantine loophole with Canada that allows for day-old chicks to come down to designated farms in the US. So it's pretty much a leaky border. And in fact, when you look at the risk of the virus spreading through migrating birds in Alaska, it's 2 bird days. So it's 2 to 3 orders of magnitude lower again. So the government were wrong, and we published this paper, and the next day we got a phone call from the Government Accountability Office, and I thought, this is serious, they want to tell us our study's a load of rubbish. But actually they wanted the data, they used our analyses, and they've reassessed the way they do surveillance programs for avian influenza. So I think that's a good outcome. Okay, but this is quite easy to predict. You just need the numbers, and you can then work out how this virus is getting about. What about these diseases that are they've not quite gone pandemic yet? They're the ones we really want to stop before they go pandemic. It's no good counting the chickens after they've hatched, is it? And here we're talking about diseases like Ebola. Can we predict the next Ebola outbreak? Or in this case, Nipah virus. Now, Nipah virus is a virus you've probably not heard much about. Thankfully, it's a very nasty virus. It causes 40% of the people that get infected to die. There are no drugs, no vaccines. And it's a very curious virus. And I got fascinated by this because I was working at CDC in the labs down there during the outbreak, just by a bit of good or bad luck, depending which way you look at it. And it's fascinating to me because why would a bat virus emerge in Malaysia and then start to spread in people? And in fact, even weirder than that, this is a fruit bat that feeds on fruit. Huge things with a wingspan about this big. And they fly out at night and they feed on mangoes. And the virus got into pigs after it moved from bats, and then really took a raging infection in pigs and then got into people. So how does that happen? It's a very weird thing. It's pretty devastating too. As well as 40% of people who got infected dying, it meant that the Malaysian government had to wipe out about a third of their pig industry, losing jobs, losing exports. So again, costly. So to find out how this virus emerged, that was our next step. Because if you want to predict something and stop it in the future, you need to know why it happened now. So to do that, we had to go to the first ever case of Nipah virus. And here's the beautiful location where it was, in a pig farm here which had 30,000 pigs. And if you went there just after the outbreak, they killed off all the pigs. But you'd see these pigsties here with mango trees hanging right over. Now this agent is what's known as a Category 4 Biosecurity Level 4 agent. That means if you want to handle it in the lab, you have to wear the big spacesuits with the helmet. You've seen this on TV. So our first question was, how would a bat virus get into a pig? And we imagine bats feeding at these mango trees, and bats are very messy eaters. These fruit bats spit out bits of fruit, they urinate all the time. So perhaps it survives in fruit. So we did our first experiment. I didn't personally do this, but we got our colleagues in Australia who work in these BSL-4 labs with the spacesuits. I think did the weirdest ever experiment in a BSL-4 lab. And we infected mango, lychees, and pawpaw with this virus. And you can sort of imagine these guys, you know, with the sort of Darth Vader suits. Oh, pass me the mango juice. You know. No, don't drink it. Anyway. The question is, can this virus persist in fruit juice? That's the big question. And what we found was surprisingly long. It's pretty resistant. It loves this sort of milieu. And here we have survival curves for pawpaw, lychee, and mango. And we actually published this study in a virological journal. One of the weirdest studies perhaps ever done, I'm proud to say. So then we have a mechanism, but it doesn't really tell us. It tells us how it got into pigs, but it doesn't tell us why it got into pigs there and then. Why Malaysia? Why 1998 when this virus first emerged? Why not in a decade or a decade earlier? So we needed to analyse this to find out what happened. So we had two hypotheses. The first idea is that bats are very sensitive animals. They don't like smoke and they fly away from smoke. Now every year in Sumatra, so this is where the index case for Nipah virus was in Malaysia. This is Sumatra, and these red dots are fires. People set fire to the forest, they burn the forest down, and they grow agricultural crops. And when they do that during a drought, you get these big fires and big haze event, and you can see the smoke flowing over to Malaysia. And it's well known that bats fly away from this smoke. So one theory was that bats that had Nipah virus flew across to Malaysia for the first time during the really big El Nino of 1997. And here you look at some NASA data. This is particulate matter, soot, in the air. And there's a huge spike in 1997. And if you look at when the outbreak was, these are the first few cases that we knew about at the time. They coincide quite nicely, so that's a good idea. Maybe this smoke forced the bats into Malaysia. So how do you test that idea out? Well, the first thing you do is you go to Malaysia and you catch some bats. This is a bit like, the recipes in the old days. First shoot your pheasant. So how do you catch a bat that's, 3 foot across and weighs 2 kilos and happens to have very big teeth and a nasty virus. Well, obviously I don't do that part of the job, you know, that's my disclaimer. Not because I don't want to, it's because I can't climb the trees. So we hired a couple of, this is an English guy who likes to climb trees and handle bats, and an American guy who likes to do that as well. And this is what they do. They stand at the top of a tree, they winch a net up, and they catch these huge bats as they fly out every evening. They winch the bat down and they put it in a little pillowcase, and we keep it over here. When the dawn comes, we take blood, we test the blood, and we release the bat. And what we're trying to say is, are these bats infected with Nipah virus? Because if they are, then it means it didn't just come over that time for the first time. Now, this obviously is a lethal virus, so you've got to wear some protection. And you've got to be very careful. You're in the jungles of Malaysia, so if you're in a mangrove forest, make sure you don't leave your equipment out at the wrong time of day, perhaps when the tide's coming in, for instance. And we had one group, and this is Raina Plowright, who was working in Australia on a similar virus, and you can see here the pillowcases with the bats in she's proudly displaying. Well, we found out that there the bats always live above water and fly over these billabongs, they call them, every night to get away. So the only way to catch them is to have one of these little tin boats with, here's the net across here. Only one of them can stand up at a time because the boat's a bit rocky. So you've got a lethal virus, a large bat with big teeth, in a billabong on a boat, which is not very good. And then we found out that in the billabong lives a 15-foot croc. So it's exciting work, I think, is one of the messages here. Anyway, what we found was, we looked at every colony in Malaysia, and these are little asterisks, and this pie chart tells you how many bats are infected, so half of them, about a third of them. And what we found was that the virus is there all the time. It circulates normally, it's part of their everyday life. It's a bit like herpes, cold sores, or the common cold. It doesn't really do much to them. So it's a natural part of their viral fauna. It just happens to be lethal in people. And these bats are cute as well, I should point out. What we wanted to do at this point is to say, do bats fly across from Malaysia to Sumatra all the time, or do they only go during the El Nino? So we put these little satellite collars, well, they're not little, but they're quite big, satellite collars that beam a signal up as the bat's flying, and then you can track it on your laptop. So the question is, and there's John Epstein releasing one of the bats to show that we do actually set them free. The question here is, in our first bat, who was called Colossus, a really cute one, does he fly across here, or does he stay where he is? And this is what happened. So you'll see the first bat flies across here, saunters around. These two bats, they live together, then they go apart for a few days, come back together to drink mango juice here in the sunset. These bats keep flying across. So basically, this whole range is their hometown, and they fly around following the smelly tropical fruits like durian, if any of you have ever eaten that, and just carry the virus with them. It's a natural part of life cycle. So if this has been going on for a long time, then it's nothing to do with the bats. There's something else that must have changed that made this virus emerge. So now we wanted to look at the pigs. Maybe there's been a change in the way Malaysians have pig farms. Have they increased the number? Have they changed the structures? Now, we can't go back in time and recreate the pig farm. The pig farm's gone now, so we can't look at how the virus would move through this population of pigs that's now gone. So what we have to do is recreate it on a computer, and this is where we call in the CSI team, the intelligent geniuses who work in mathematics, who can simulate an epidemic on a laptop. And what we're trying to understand here is, how did Nipah get into this pig farm and persist for over 2 years? This is the first human case, and then here's the big outbreak. It survived for 2 years in this pig population, which is really unusual. If you imagine a virus getting into a population, if it's one that's so virulent in pigs, it should infect all the pigs. Some of them are going to die, the rest become immune, and the virus has nowhere to go. But this one persisted for 2 years. Something weird happened. So what we did is we went back to the index farm. We met with a manager. We got the logbooks with all the data in. We interviewed people in Malaysian pig farms. It's not a pleasant job. They're very smelly and horrible places. And Juliet Pulliam and Andy Dobson created this mathematical model, and basically each one is a different type of pig. They're sows, piglets that they give birth to, growers that then have fattened up and become pork sausages here. And some of them go back into the farm and recycle. But this is a very structured, intensive production. So this happens very rapidly, and there's a very high level. And what Juliet did is write equations that describe the viral dynamics in each of these age classes, and then ran a simulation on a supercomputer to look at how the virus would behave. And what we're trying to understand is, how does this virus persist for so long? Now, if you ignore all the wavy bits and just look at the green bit here, that's the infected pigs over time. So on the computer model, you introduce the virus, you get an infection, and then because the virus runs out of animals to infect, it dies out. And whatever we did to the mathematical model, we couldn't get the virus to persist. And then we heard about this bit of research that had been done that shows that if you analyze the virus from the beginning of the outbreak to the end, it shows that it's changed. Now, maybe it evolved, or maybe bats repeatedly introduced the virus into these pigs. So we tried that in the model. We repeatedly introduced the virus, and suddenly you get a completely different dynamic. It goes up a little bit and then persists for a very long time, just like it did in the outbreak. Now, the reason for that is, if you imagine that this epidemic is a bit like a fire. You know, when you've got a fire and you throw on a load of wood and it rages up and you feel nice and warm, it rapidly dies out. It runs out of fuel. But if you throw some damp logs on, it sort of keeps the fire going. Now, what happened here is the pigs that were initially infected, the female pigs gave birth to piglets that carried the maternal antibodies, just like children have the parents' antibodies. Eventually, they lose those antibodies over time. So they're the damp logs that gradually lose antibodies and become susceptible again and get infected and keep the dynamic of the virus ticking over. So we had a very weird situation with bats feeding on mangoes that are planted around the farm, and then this rapid intensive production leads to an outbreak. If you look at pig production in Malaysia now, they want to start again. You can see over time this has increased, and then here's where Nipah virus emerged, and they culled off a third of the farms. And by the way, look at mango production. It goes up almost identical and then drops. When you kill off the pigs, you also lose mango production because they plant things side by side. So the Malaysians want to start up again, and we work with them to say, let's look at our mathematical models, let's look at where pig farms are, the size of the pig farms, and where bats are, and what fruit trees you've got. And what we showed them was that, yes, you could do this pig farming again, but you wouldn't be able to do it here where the bats are. You're going to have to do it somewhere else. There's a high risk here and a much higher risk down here of people getting infected. So we now have a way, through this few years of work we've done, to help Malaysia with its economy, but also to prevent future outbreaks of this pretty nasty disease. Now, those bats don't just live in Malaysia, they live all across the tropics in the old world. And if you look at one place, Bangladesh, there's been a repeated series of these outbreaks of Nipah virus. And here it's even worse. 70% of the people that get infected die. Now, these aren't high numbers, but these are only the ones we know about. It's a very nasty virus. And what's really worrying is in Bangladesh, it's changed. There's a different ecology. In Bangladesh, people put little taps in date palm, and it's a bit like maple syrup collection. You can see a pot here. And they shimmy up these trees, they tap a little tap in here, and overnight they collect a nice jar full of maple syrup, date palm syrup, and they sell it off in the morning to passers-by. I've not drunk it because I know it's not a very pleasant thing. What happens is bats come here and lick at this juice, urinate into the pot, and they carry Nipah virus. So when people are drinking this, they're drinking a Nipah virus culture. And we know that the virus can persist in fruits, in fruit juice, in sugary fluids. So this is, it's a very weird thing. It's very interesting, but it's also extremely sad. This is a guy that we've got to know quite well. He's a date palm gotcha, they call them. They're the ones that collect the date palm, and he sells it. And, he's had a hard life. He infected his family. 3 of his family members got infected from date palm juice he collected. They died. They infected their aunt. She died. She infected the local religious leader who came round to visit her when she was dying. when he was dying, people came around from miles about and visited him, touched him, hugged him, kissed him, and they got infected. So it led to this huge outbreak. It's a foodborne infection with now 5 cycles of human-to-human infection. So here we have the virus starting that first stage of pandemic emergence. And this is the religious leader, patient C, and there's the big outbreak that followed. Now, what we're doing, thanks to a new grant we've just got from National Institutes of Health, is working in Bangladesh for the next 5 years to try and understand the dynamics, try and do something about that. Now, our group's also worked on SARS. We found that SARS also has bats as a host. SARS emerged in China as part of the food industry. It's a restaurant trade disease. Unfortunately, in China, people eat live animals. They keep them live until the last minute, and they eat a wide variety of wildlife. And they also eat bats. This is one of the markets where you can buy these live animals, and they butcher them there and then on the floor. This is not a very sanitary place, and if these animals carry viruses, it's a great way for a virus to spread around. What we found is that these bats that are in cages, these cute little kids looking at some bats, he finds it fascinating. You can buy them for food, and they happen to carry this SARS virus. I don't know why you would want to eat one of those things, by the way. That's a pretty ugly lunch. And it's probably a lot better out in the environment. Bats are great insect eaters and pollinators. They need to be out there in the environment. So why did SARS emerge in China from bats? Well, if you look at China's economy relative to the world, it's been growing at a huge rate. Even with the downturn, it's now projected to drop to 6% per annum, and that's considered bad. So the economic boom in China means that a lot more people who eat very traditional things have a lot more money to spend on them. So there's been an upsurge in food production. This is the building site for the Olympic Games. You could have gone there just before Olympics like I did, see all this amazing technology, rapid growth, and then here's a guy pulling out old tree stumps out of the rubble, taking them off and selling them as firewood in one of the villages in the outskirts of Beijing, taking them by donkey and cart. Incredible rapid growth. And here's a restaurant that when I first went to China I was taken to. It's a posh restaurant, and I went in there and everything's live. Here's some snakes you can buy. You pick your snake, they kill it in front of you and you eat it. If that snake has a virus, there's much more chance of infection happening if it's kept alive until the last minute and chopped up with more people around. So that's why SARS really emerged in China, this economic boom. I think, again, it's telling us that disease emergence is a product of what we do on the planet. Just like Nipah virus emerged due to intensive agriculture, SARS emerged due to growth in economy in the restaurant industry, eating wildlife. It's a product of what we do. It's part of our cost of doing business on the planet. And I think what we need to do is to start to deal with this seriously. So here's how I propose, and I want to finish the next few minutes just to talk about the ultimate prediction for emerging diseases. How can we predict the next big one, the next HIV or the next SARS, the next West Nile virus? Now this is a big challenge, and I want to do this because I find it fascinating, but also because there's a well-known virologist called Fred Murphy says you just can't possibly do it. So that's a good reason to give it a shot. Now he does have a point. It's going to be very hard, if not impossible. And this is why, because what we really need to do is to say we need to predict the unknown. We don't really know how many viruses there are out there ready to emerge. We know 2,000 viruses we've discovered so far from wildlife. Now, if you look at the viruses that come into our population, usually emerge from vertebrates. Now, there are 50,000 vertebrate species. If each of those has 20 viruses, let's say, and that's a low estimate, because humans have well over 40 viruses, or more actually. If each one has 20, that's 1 million unknown viruses. We know of 2,000, so we underestimate diversity of viruses by 99.8%. In fact, bats, we know there's all these weird viruses coming from bats. Bats alone have, according to this, 20,000 new viruses waiting to be discovered. So that's a problem. And getting to understand that unknown is very hard. And And this is what we tried to do. So what we did is we built a database of every emerging disease. We went back to basics. Let's go from what we know about them. Let's study what we know. So we know there's been about 450 emerging diseases in the last, say, 6 decades, since 1940. We did a couple of years' research to try and identify the original site for every one of these diseases to emerge and the reason why they emerged, the time and the date and the place. And then we plotted it on a nice map for you here. And as you can see, where I'm from, England, is a very bad place for emerging diseases. It is actually why I moved to the States. I moved to New York, which, is a very bad place for emerging diseases. But of course, this is a biased map, because what this really is where these diseases were found. Now, if you have more people looking for them, you're going to find more. And these countries, Europe and North America, we can afford more researchers, more hospitals. So how do you get around that bias? And what we did is we had to, and I'm using the royal we here, it was actually an intern who did this miserable task of searching through 6 decades of a journal and plotting the geographical coordinates of each of 17,000 authors into the database. And then we correct for where the researchers were working. So that gives you a measure of where the surveillance effort is. And once you balance that out, you can start to see the true patterns. And also, we've increased the number of researchers over time, so there's been a bias that way. Now, doing this approach, we could actually start to answer some really big issues. Are emerging diseases a real issue? Are they really on the rise, or am I just building up a bit of hype? Well, you can see from the raw data that decade by decade, they're increasing. And this is the 1980s with HIV, and HIV alone caused a little blip because it caused a bunch of new diseases to emerge. But you have to correct this for the number of scientists increasing over time. And when you do, you still find a significant growth. And what's really worrying is if you look at the yellow bars, they're the diseases that come from wildlife. They're increasing over time, and they represent more than half of the pathogens that emerged in the last decade alone. So zoonotic diseases from wildlife, SARS, HIV, Ebola, they are a real issue. Now we can test, using this database, a couple of big ideas. Number one, are diseases caused by human activities, like I've been telling you for the past few minutes? Well, if that's true, the risk of them emerging should correlate with where human density is on the planet. If, as I'm saying, that the wildlife diseases that spill over to people are the big risk, well, where are they? Where do they correlate to? Shouldn't they correlate with where wildlife are? If every wildlife species has 20 new viruses, isn't that where they're going to emerge next? So we got some data, and we plugged it into an analytical regression and looked at the correlations between these factors. So if you look at the zoonotic diseases from wildlife, the SARS, the HIV, it's the only group that really correlates with wildlife populations. Every group of pathogens is strongly correlated to human population density. They are a product of what we do on the planet. And because these data are spatially modelled out, we can actually produce maps now of risk. And what we want to see is, instead of England, where I'm from, being a big risk, or New York, shouldn't the risk for new diseases start to spread now to places like Central America, where all the wildlife are? Shouldn't it be a product of where people and wildlife come into interaction? And sure enough, when you plot it out, that's what happens. Instead of the dominant risk being in Europe and North America, that's still a risk because we change the environment so much. But now you start to see Southeast Asia, Bangladesh, where Nipah emerged, Malaysia, Africa, where Ebola and HIV emerged, and Latin America start to become hotspots. So now we have a map of where the next HIV is going to come from. It's very rough, but it starts to point to where we should be looking. Are we looking there? You'd think we would be, wouldn't you? Well, if you plot out the number of researchers and where they're from, it turns out we're not. We're looking in the wrong place. Surprise, surprise. The richer countries in the world look locally. You know, we have a whole team of people doing bat rabies surveillance in the States. Well, that's no good if HIV emerges and kills everybody, is it? Or pandemic flu. What about the hotspots? The lighter the colour, the less people looking. And I think we're really missing the boat here. So the big message is we need to reallocate a little bit of our effort to the right places so we can stop these diseases. How do we do that? Well, our organisation, a bunch of other organisations are starting to do this. And these are some of our field sites. And we're working with local scientists, local labs to do wildlife surveillance, to look for new pathogens before they emerge. How can you find an unknown pathogen? Well, there's a bunch of new techniques. And this is Ian Lipkin at Columbia University, who specialises in discovering new viruses. And what you do is you look for the DNA or the RNA of a known virus and you change it a little bit and look for the RNA of something that's somewhat similar, i.e. its sister or brother virus that we don't even know about yet. And we've already tried this technique out. These are some of the bats we used. 8 bats from Bangladesh, pregnant females. Everybody knows when you're pregnant, you're immunosuppressed, there's more chance of a virus that you've got being present in the tissues. We took blood, we tested them using his technique, and we discovered a new virus already. So I think this is a valid way to seek out the next HIV or SARS. The next big question is, do we have the skills to stop it before it emerges? Well, I think that's just a matter of working with people who are at risk, who live in these countries, who are exposed to things like date palm juice, pig farms, bushmeat, and explain the risk and say, you know, you're putting yourself and your families at risk as well as the rest of the world. Try not to do that, mate. That's the strategy. Hopefully it'll work. So I want to just finish by thanking all, I mean, obviously I can only do some of this, but working with these fantastic colleagues who I've been working with for the past decade or so, especially John Epstein, who runs the Malaysia project, Mark Kilpatrick, who runs the West Nile virus program, Steve Luby in Bangladesh, Juliet Pulliam, who did the modelling, and various other people, Kate Jones, who helped with the hotspots analysis. And of course the funders, without whom you can't do any of this work, and the wildlife, who put up with a lot of horrific things just so we can understand something that they're not really responsible for. So thanks very much for listening. I look forward to questions.
Thank you, Peter, for a very enlightening talk. I'd like to, as we move into question and answer period, request that if you have a question, please come up to one of these microphones so that we can hear both the question and the response. Thank you.
And you're not allowed to leave until you ask a question. If you whisper loud, that's considered a question. That's the...
I'll ask my question. I'll just yell. I have a big voice. So something I think is just kind of relevant here in Montana. So obviously the world population is increasing, but it's not, I think, just sheer numbers of distribution. And here, one of the things that comes to mind for me is that we're living in almost an urban wilderness interface. And I wonder if you can speak a little bit to the effect that we have of just directly interfacing with the natural population. As opposed to crowding animals together, et cetera.
Yeah, well, it's interesting. I mean, you think you're doing everything right by living out in a beautiful place like Montana, that you could not possibly be at risk of disease. But of course you are, and it's really interacting with wildlife and livestock that is the big risk. Now, we were talking earlier about ranching of deer and elk together. What happens when we crowd animals together is we increase the potential for a pathogen to move across from one animal to another and build up its numbers and then start to become a risk for us. So I think we've seen that with mad cow disease, we've seen that with chronic wasting disease, and yes, it is a risk. And of course the classic disease for Montana and around here is brucellosis, which has become such a political issue. It's quite amazing. It's really a disease of cattle that came over here with cattle that we brought over from Europe and then got into bison, and now we kill bison because they're infected or because we think they might be infected. It's an incredible situation to be in, but really we are in this urban-rural wilderness interface, and that really is the thing that drives these diseases to spread.
Two quick, I think, one and related to the question already, is one is as pressures of population moving into tropical areas, the agricultural development and bushmeat and all this, and increasing populations. I assume that that is a factor in increasing the risks for viruses being spread in a either localized or more pandemic kind of way. But the other factor here is global warming, which is changing also possibly species and their locations, and that stress to the environment and to the risk of these kinds of diseases. Is that something either you or who is doing that kind of, you see things about the effects of global warming on potential for pandemics. Where is that work being done, and what can you say about that?
Well, thanks for that. Yeah, you're absolutely right. We've actually just started work on the question of how will those hotspots change with global warming. Now, it's pretty clear global warming is happening. I mean, I was not a great believer in this until I saw the data. And if you look at the trends of earlier flowering of plants, earlier migration of birds, especially melting of glaciers, it's pretty obvious. And I know that we're talking today about mosquitoes living longer in Montana. That's bad news for you guys. We've got plenty of them in New York. You can have some of ours. What can we say about what will happen under global warming? Well, two things. First of all, what happens when you change the climate is you force animals to migrate. They want to move to the climate that they're used to, and they will move, and they'll move up a mountainside, they'll move north or south depending on the temperature envelope. When animals move from one place to another, they come into contact with other animals, other populations, other species or livestock, and pathogens move. So a general prediction is very straightforward. Pathogens will emerge at a higher rate as we change climate. That's pretty obvious. We are starting to do some analyses on that, and there are lots of people out there looking at diseases that may emerge due to climate change. One of the most commonly cited of these are vector-borne diseases that are transmitted by mosquitoes and biting flies, for instance. Vectors that depend on water and temperature and climate for the production of larvae. So if you get hotter conditions, you get more larvae, you get more rapid biting rates, and you get higher infectiousness of some viruses. Now, West Nile has actually evolved since it came to the States. It's changed into a new strain of West Nile virus, which is more virulent, it develops more rapidly in mosquitoes, and is therefore more of an issue. And there's a lot of discussion over maybe that's in relation to climate. And it's certainly better adapted to future climate change in the US. So I think we're in for more of the same, and some surprises too. So, you can't stop global warming, but not rapidly anyway, but you can certainly get ready for what's going to happen and try and deal with it. Now, the other point that you made was that this is all related to population increase, and that's absolutely true. The bottom driver of many of these things is global population increase. And of course, that's gradually beginning to tail off, but that also is not going to change in the near future.
That was a great speech. I had one quick question. I'm curious on the country of origin labeling and whether that's going to increase or decrease the expand and to trace. What's your impact on the movement of tracing and finding the parasitics through that country of origin labeling we have on packaged meats that we currently have in the United States?
Well, yeah, I mean, what's incredible to me about that issue is that there's so little done in terms of, right now, in terms of tracing trade that could allow pathogens to get in. Now, yes, meat and meat products are tested for a number of listed agents. And knowing the country of origin means you're in much better situation for looking for the right types of pathogens. And I think we're doing the right thing, We've got to take this issue seriously. It doesn't only cost us lives, it costs us money too. And if a trade is there to make money, it's pretty dumb to not protect it. It's an insurance policy. But apart from that, what are we doing about wild animals coming into the country? We just finished a study where we To get the data, we had to file Freedom of Information Act requests, which is pretty crazy that we've got to do that, for Fish and Wildlife to give us the number of animals coming into the US every year. And we got every single animal coming in through every port of entry. And between 2000 and 2005, there's 1.6 billion live animals coming in. How many of those are tested? Probably less than 1%. How many pathogens are they tested for? I think about 5. You know, there are only a few notifiable pathogens of wild animals that you have to go out and test for. So this is a huge issue to me. And it's not that this is just a bit of scaremongering. There really are diseases that emerged here due to the wildlife trade. Monkeypox is one of them, for instance. So I think that's a wide open issue that needs to be addressed. And if we think about why did West Nile virus get in, it got in through aeroplane travel into New York, bringing things into the country. Why aren't we in those ports looking for the next one? Not just testing for a handful of disease for cosmetic purposes. So I think this is a big issue that will become part of legislation in the future. I hope so anyway.
Why bats?
Yeah, that's a good question. I feel really sorry for bats. They've got a bad rap. You know, some of them are ugly, as I showed you. Some of them are beautiful. Most people don't like them. They don't fly into your hair, but they are kind of weird. They do carry rabies, which is the only 100% almost lethal disease on the planet. I think bats don't carry, if you do the analysis, they don't carry an unusual number of pathogens. It's just that over the last decade, a bunch of them have emerged. Now, there's a lot of work going on at the moment saying what's special about bats, but I think the reality is what's special is we've started to get in their territory. In Malaysia, they're putting pig farms in fruit bat habitat and planting fruit trees. You know, you're going to bring the bats in. In China, they're eating them at higher and higher numbers. I think eventually as we reach out and encroach around the planet, we're going to come across these unusual wild animals and start to get their infections. The reality is bats are 1/5 of all mammal species, so there's a lot of them out there, and we'll probably see some of the other 20,000 viruses that they carry emerge in the next few years.
Thank you for your presentation tonight. On NPR earlier this evening, you talked a little bit about bioterrorism, and you focused on something just long-term persistent impacts, economic impacts through agricultural disease. I was wondering if you could elaborate a little bit on that.
Well, certainly. You know, we've seen a huge effort over the past few years with the current administration on biodefense, as it's called, which I think is a good thing. We did have a real bioterrorism attack with the anthrax letters that were sent out. Thankfully, they've got to the bottom of that. You know, one thing that we underestimate is the potential for some of these other pathogens. There has been some discussion of this at the National Academies, for instance, on agro-terrorism. Are there some pathogens that bad people could put into pig farms or poultry farms or cattle farms and, you know, cause a problem? And you think about the impact just in terms of the scare of one infected cow with mad cow disease. I think this is a big issue, and it's certainly something that's starting to be looked at now, but is right now under-researched.
Do you think that globalization and increased human and animal interface is going to increase the amount of cases of antique diseases or diseases that we thought were cured, or the reserves fell down, but environmental change raises the reserves back up?
Well, I really like your turn of phrase there, antique diseases. This almost gives them a nice image. But yeah,
like the re-emergence of trench fever.
Absolutely. And it really is an issue of globalization. Now, you know, I'm a globalizationist. I'm part of the problem. I fly around the world. I eat things from weird countries that I probably shouldn't do. It certainly does drive disease. The evidence is there. As we've increased our travel and trade, we've dragged in these diseases from other areas. And as we do more of that, we'll see not only new ones, but also some of the old ones re-emerge. And not only that, we'll see drug-resistant versions of the old ones emerge. So yes, that's a serious issue for sure.
Most of your comments on these emerging diseases focus on the animal side of things. We know that in the developing world, there are big problems with drinking water, sewage, and so forth. Would you say that side of the equation should also be taken into account?
Yeah, that's a good point. I mean, yeah, I work on emerging diseases, so I'm going to tell you they're a big issue, aren't I? Now, there are diseases out there. The biggest killer, for instance, is diarrheal disease. Malaria is still a huge problem globally. I think what I'm trying to say here is, we know the solution to malaria. That's malaria tablets. It's mosquito control. We don't know the solution yet to the pathogen that's going to emerge that's going to infect 40 million people like HIV. So I think we need to get ready for this. But at the same time, I take your point exactly, that I work in Bangladesh. One of the biggest problems there is hygiene. It's potable water. It's sewage. Avian influenza is a big threat. But one of the great ways not to get infected with influenza is, people worry about airborne transmission, but actually hand washing, you get influenza from touching surfaces, and you get other diseases that way too. So hand washing is a key issue. And these basic programs will protect us not only from old pathogens, but from new ones too. Very good point.
When you model human to human or animal to human transmission, how much of a factor or change in the equation is there when you have a intermediary, like a mosquito vector or some other kind of vector?
Yeah, it changes everything actually. And the reason it does that is because it gets around a lot of problems. A disease with a vector is actually more of a difficult disease to control for a couple of reasons. One is that it's got an extra animal to hang out in and persist in. So if the host population drops really low or they move away, it can persist in the mosquitoes for longer. Also, it becomes what's known as frequency dependent. Now, what that means is that it doesn't depend on this density that a lot of these diseases do. Mosquitoes bite repeatedly over a period of time and can infect a lot of people. And it's irrespective of density. They'll go out and seek you down and hunt you down and bite you. That's what they're designed to do. That's what they've evolved to do. So, yes, they're a worrying issue, and you have to completely change the models and the approach. And that's why I say something that is a flippant comment, but West Nile truly is here to stay in the US. It will be almost impossible to eradicate. And, it's in over 100 bird species. It's in over 20, 30 mosquito species. It's now endemic, and every time we introduce one of these pathogens, especially the vector-borne ones, it becomes a real issue to get rid of.
Well, let's thank Peter again for enlightening